My name is Vitezslav Kucera, and I want to talk about to my big interest now in my PhD studies, which is combinatorial recombineering and minimal cell, and how this recombineering can be used for a directed de-evolution of the minimal cell. So what I'm talking about when I'm talking about the de-evolution or minimizing of the minimal cell, because minimal cell is already minimal, but it is minimal in the term of its functionality. So when you have the normal cell, it's got a lot of these genes that provide the function of the life. And in minimal cell, you reduce these non-essential functionalities of normal cells to a minimum. But what we are interested in the lab is a minimization of a different kind, which is minimization of composition of the cell, and specifically the minimization of the proteome, so the proteins and their actual composition, and specifically minimization of amino acids. So it's similar to the minimization in function, where we have these 20 canonical amino acids that all living cells do share. But what we think, because we are a lab interested in the origins of life, we really think that the early life did not have all these 20 canonical amino acids when it first emerged. But it is very hard to experimentally somehow prove this. So we are interested in reducing one or more of the amino acids from the proteome to get near to that state with minimal composition for proteins, because we know that proteins can definitely function with fewer than 20 canonical amino acids. We even know that they can at least have structure and sometimes even function with 10 amino acids. So for that, we chose SYN3, which is a great model for that, because it already doesn't have a lot of amino acids in terms that it's got only a few hundred genes, which means that a lot of the amino acids are already reduced. So it still uses 20 canonical amino acids, but it does have fewer of them. So if we would want to do that, currently most of the options include synthesizing chemically the genome of the SYN3, or editing this genome inside yeast and then transplanting it into the chassis of some cell and seeing if this minimal composition genome can work. But what we are almost sure of is that this would not actually work, because it's such a drastic reduction that probably there would be no viability. Most of these proteins would probably not even work. So we want to approach this from a more top-down approach and gradually decrease the composition of the proteome of the SYN3. And for that, the best way to do that would be for us an iterative combinatorial engineering, so genome engineering. So we take the SYN3 and iteratively we remove or replace the amino acids inside the genes, some selected amino acid that we will choose one by one, see what effect it does have. And ideally, by the end, if we can do this for all the genes, we should come up to either a cell that really does not contain all of the 20 canonical amino acid, or at least some intermediate that will be minimized in the composition of the targeted amino acid. For that, we have one problem, and that is that genome engineering in SYN3 is actually not something that can be done easily. So for that, my plan for PhD, apart from another project, is to introduce an oligonucleotide recombineering inside SYN3. So why oligonucleotide recombineering? It's only a very, very simple genome engineering tool that can be utilized in many cells, and that is because it's mostly reliant on genome replication, which is something that all cells, including SYN3, does, which is already very helpful. And so in this recombineering, you put mutagenic single-stranded DNA inside the cell, and if you design the mutagenic DNA well, it will anneal to a single-stranded part of DNA of replicating cell. And if there is a mismatch, this mismatch of the single-strand DNA can be then incorporated inside the DNA of the new daughter cells, and in 2 generations it can be completely incorporated inside the genome as a novel mutation. There are a few problems with that. First one is that this protocol relies on mismatch repair not being present in the cell, which is also great news for SYN3 because SYN3 has most of its mismatch repair removed already. It's got few DNA repair enzymes, but these have been shown to also not hinder the recombineering. So the recombineering mostly relies on the mismatch actually not being present. So this should be something that is already done in SYN3 for us. And so the main problems that we will actually be solving is the interplay between the single-stranded annealing protein, or SSAP, and the single-strand binding protein. So what are these? I will start with the single-strand binding protein, because that's a quite simple protein that also almost all organisms, or as far as we know, all organisms do have this protein, because when the organism replicates, the naked single-stranded DNA cannot be just left there naked, and for that the organisms usually have these single-stranded proteins, single-strand binding proteins that can bind this DNA. We know from the proteomics of SYN3 that JCVI SYN3A does have the protein 26, which should be a single-strand binding protein that binds DNA during replication. And the good thing about this protein, which also makes us hopeful, is that it has got a 40% sequence identity to a single-strand binding protein of Bacillus subtilis. This sequence identity is quite important, or we hope to be so, because this single-strand binding protein, for the recombineering to work, needs to interact with another protein, and that is the single-strand annealing protein. These proteins are usually viral recombinases, so they are derived from phages that infect bacteria. And what they can do is that they can take a single-stranded DNA that is present in the cytoplasm of the cell, and they can direct it directly inside the replication fork of the replicating cells. During this, they interact with the single-strand binding protein, and that's where there need to be a match between the single-strand annealing protein and the single-strand binding protein. So if we can find a single-strand annealing protein that will work well with the single-strand binding protein of SYN3, this should, in theory, allow us to actually edit the genome of SYN3 directly inside the cell, not relying on the ex vivo genome engineering that can be done now. So for that, we will be screening a lot of SSAPs for their activity inside the SYN3, and hopefully, if we find some that is active, we can proceed with that further, and this will in turn allow us to actually do all of these changes inside the proteome of SYN3, and in the end, we will either end up with a cell that is completely reduced on the biochemical level of proteins, or at least is partially there and will show us something about the biochemistry and protein structure of both minimal cells and also the rest of the cells. So with that, I want to thank you all, and also big thanks goes to the Laboratory of Synthetic Biology, where I'm doing currently my PhD, and to all of the amazing people there and all of the collaborators that work with us.
Have you guys, Vitezslav, have you guys used the landing pad to introduce? Have you tried introducing genes to Syn3B yet?
Not yet, not yet. I sadly am just now starting to work with it, but it is currently something that we want to do. Either doing it with the LOXP, the Krelog system that is already in the Syn3B, or also with the transposases that I think there's a protocol for transformation of Syn3 with transposases. So that's also something that we can do.
You should have the plasmid for introduction in the LOXP site, and of the two protocols, the plasmid approach is in the LOXP site is definitely easier.
Okay, yeah. Thanks. I think we have it, so I think we were planning to do that in the following months.
And my team will be happy to help you, advise you about this. So feel free to reach out anytime.
Thanks a lot. Yeah.
What candidates do you have for your phage protein?
So for the phage protein, so that is the single-strand annealing protein. Currently, we are aiming for GP35, which we know this one works in Mycoplasma pneumoniae. So it's not a completely related organism, but it's very close. So we are hopeful that this one should have at least some activity. We don't know if it will be perfect, because we know that even in Mycoplasma pneumoniae, the activity is quite low, and you usually need some counter selection strategy for better selection of the mutated strains. But that is the currently the best protein. Yeah.
Okay, and if it works in pneumonia, I think your chances that it'll work in our cell are really good.